Hi students, welcome to Year 11 Biology and Module 4 Ecosystem Dynamics. This is video number 10 and we're going to be reviewing, our understanding of radiometric dating. So what we are doing is to continue to investigate and analyse past and present technologies that have been used to determine evidence for past changes. And one example of those technologies is the technologies that are applied to radiometric dating. What we want you to be able to do is to very briefly describe radiometric dating, to link radiometric dating to changes in the past, and specifically, if you can, explain how radiometric dating provides evidence for past changes. So I guess the first and the most important thing for us to do is to talk about radioisotopes. We've talked about the fact that different elements can exist in more than one form, and those changes in form are based on mass differences, specifically the number of, or the ratio of protons to neutrons in a nucleus. If this ratio is not ideal, then what can happen is that the nucleus can radioactively decay, release particles of radiation, or at least radioactivity, and therefore that atom will be transformed into a different type of atom. Now, there's a number of different elements that we can use in our radiometric dating, and the half-lives, that is the amount of time it takes for a particular half of a sample of radioactive material to decay, is different for different types of elements, and so therefore and different types of isotopes actually within those elements. So it means that we can, depending on what sort of time frame that is for the half-life, that will determine what sort of age of rocks or fossils we're able to age. So we do take geological specimens. Sometimes we take the specimens themselves if we can find evidence of radioisotopes within them. Sometimes we may take samples from above or below, particularly if they are igneous samples where there are a higher likelihood or a higher proportion of radioisotopes, and we can therefore date that particular rock sample and talk about whether the samples that we're specifically interested in are older or younger, depending on whether they're above or below the sample that we're looking at. It can help us to get an understanding of evolutionary sequences and past environments. Obviously, what we want to do is try and get a little bit of chronology. Look at the age of different types of environments. Also look at the types of fossils that we find there, and to see whether or not this idea of age tells us something about when climate changed, when different organisms appeared, perhaps super predators, or even the presence of humans, and how that may have impacted on the plants and animals that were present at that particular time. We can also do this with microfossils, and our techniques for understanding and in fact even just finding the presence of microfossils has improved considerably, particularly over the last 30-40 years. Zircon crystals are often used, and sometimes we actually look at fission tracking, and I'll talk about that very briefly in just a moment. You can see from the diagram that in any mineral deposit, the vast majority of the atoms are not radioactive, and it's only a small portion of them that are. And those that are going to release that radiation and are going to transmute into a different type of element or a different type of atom. And so therefore, by looking at the proportions and the half-lives, we can get a bit of an idea of the age of each of these different types of samples. Here's an example of some of the radioisotopes that are used to date different types of rocks. And you can see these are the ones for the very, very old, or the oldest types of rocks. So uranium 235, 238 right up there. Thorium-232 is another one that's used very commonly, and it's part of the radioactive decay sequence for uranium. Rubidium's there. Potassium-argon is another couple, a radioactive isotope couple. So the potassium 40 is radioactive. Argon 40 is the stable daughter nucleus. and also things which we haven't listed, such as carbon 14. Now, of course, the thing with carbon 14 is its half-life is around about 5,770, something like that, years. So therefore, carbon 14 can only be used for very young, relatively young fossil material, much more than about 10 half-lives. And the amount of material that remains is too small to be of any real value. So, so that's one of the reasons why we look at different types of radioisotopes for dating different ages or potential ages of different rocks. These are just a couple of the specific dating technologies that are used. The first one is the shrimp. That is the sensitive high-resolution ion microprobe, and shrimps have been used, are being used more and more now as we refine the techniques to look at this sort of high-resolution material data that we get. Fission tracking data is also something that, and I guess if you're looking at explaining fission track data and luminescence data, you're looking at similar sorts of things. You're looking at the fact that often when a nucleus decays, the daughter nucleus remains in place. Nucleus remains. But the radioactive product can kind of push its way through the material, and you may even see where that particle, it could be an alpha particle, beta particle, could be gamma radiation, and that sort of has an impact on the crystal structure of the rocks in which it's found. And so as that particle tries to escape, we can almost see evidence of where it went. And that's the principle upon which things like fission tracking and luminescent dating are based, that we can track where the particles or where the radioactivity has gone after the nucleus is decayed. So these are just a couple of different dating techniques. Obviously, you want to look at these in a little bit more detail. We just want to hit you with some sort of general terms so you understand, or at least that you've seen some of these before, and we'll look at them in a little bit more detail in class. Thanks for watching.